This is an introduction to the seed plants. We've just finished talking about the seedless vascular plants, and so the seed plants are going to have some similarities with those seedless vascular plants, namely the vascular tissue, the dominant sporophyte, some other things, but they're going to have some really big differences in their life cycles, and I want to talk about some of those key differences right now. As we do that, before we do that, we're going to kind of review what we've seen so far. So the first thing to think about is sperm and how they get where they're going. In the green algae, we talked about sperm with flagella, so they swim. They're made on the gametophyte, they're released into the water, and they swim. No problem. In the non-vascular plants, we also saw swimming sperm, as we did in the seedless vascular plants, and I hope that you got the chance to see these fern sperm in lab, because look at this crazy thing. So you saw them, if you saw them, they were just kind of wiggling around in a circle, and we talked about how that makes sense in the environment in which these sperm are released. So in all of these groups, fertilization requires water. You can't swim without water, and this then restricts the range of the organism. They have to be somewhere where conditions are at least sometimes suitable for fertilization. All right, so only places with water, extra water on the surface of the ground is favorable for fertilization in these seedless plants. Let's think about the Gametangia. Oh, I'm out of order here. Okay. In the green algae, we didn't see any complex Gametangia. In our non-vascular plants, we saw Antheridia and Archegonia, Archegonia, moss, and liverworts, respectively. You saw these, we talked about them in lecture, you saw them in lab. So multicellular complex structures to hold on to and protect the gametes, the egg and sperm. We also saw these in the ferns. I had at least a picture for you printed out in the lab. You could have seen these if you looked really closely on the fern gametophyte that you made a slide of. Even if you didn't see them, you're seeing them now. We have complex multicellular structures that hold on to our eggs and sperm. So here's an antheridium with lots and lots of sperm in there. Here's an archegonium, same sort of arrangement as we saw in our other organisms, cells surrounding the egg cell, which is right here under my laser pointer. All right. So what happens in the archegonium specifically is fertilization, and this is then where our new sporophyte starts out. And in all the land plants, we see the sporophyte dependent on the gametophyte, at least to begin with. So we also talked about how this requirement of water for fertilization restricts the ranges of the sporophytes as well as the gametophytes. Okay, get ready for something new. The pollen grain. This is something that we have in all of our seed plants. And you'll see, we'll talk about now, how this frees plants from this dependence on water for fertilization. So get ready. The pollen grain is the male gametophyte, the multicellular haploid generation. It's coated in this molecule we've heard of before, sporopollenin, which makes the pollen grain resistant to drying out. It protects the goodies that are on the inside. We'll talk about what they are. Here we go. So, the structure of our pollen grain, and we could write a whole book about this. We're going to talk about this in a really simple way. We've got the outer layer, the exine, we call it, EX, refers to outside. The exine layer, this has the sporopollenin. We have inside two cells. We've got a cell called the tube cell. I drew this picture, don't you like it? The light green is the cytoplasm. So the tube cell takes up most of the inside of the pollen grain. It's also got a nucleus. And then a smaller cell is called the generative cell. So here, yellow is the cytoplasm of the generative cell. and that black dot there is supposed to be the nucleus. When the pollen grain is released, we just have these two cells. A little bit later on, and we'll talk about this next week, a little bit later on, the generative cell divides into two sperm nuclei. So you can think about the pollen grain as a little transit package for the sperm. So now, instead of our sperm swimming through water, the sperm are flying through the air, or they're being carried by an animal like a bumblebee. Okay, so now, instead of swimming sperm, we've got flying sperm. This frees the seed plants, in part, from their dependence on water. For fertilization, at least. Of course, they still need water to survive. Okay, but they've got the system of roots, a system of vascular tissue, so they can move water around their body, and they can go really deep down into the ground to find that water if needed with their roots. Okay, let's think about sporangia. In the green algae, no such thing. We have our single zygote, our diploid cell divides by meiosis, makes spores with flagella, and they just swim away. We don't have any structure that holds on to the spores at all. In the non-vascular plants, we had those terminal sporangia. In our seedless vascular plants, we had the lateral sporangia. In all cases, in these plants, we've got essentially a stalk with a capsule at the top that holds on to the spores. So the spores originate in this multicellular structure. They originate in this multicellular structure. They mature there, and then they're released from there. So in these plants and the green algae, how do we get our offspring out into the world? Well, I've already kind of told you. In the green algae, the spores are made by meiosis from the zygote. They swim. So they're released into the water and they swim away. That's how you get your offspring out into the world. The spores have the potential to grow into a new multicellular individual, the gametophyte. So the spore on its own is the beginning of a new life stage. In the non-vascular plants, in the seedless vascular plants, we have spores that we're sending off through the air. So we're still dispersing our offspring by spores, but we're doing it through the air instead of through the water. This is a great little step-by-step diagram of that spore shooting, it's called. So the action of the annulus, it pulls back, launches open, pulls open this sporangium rather, and then it's sort of cocked back like a catapult, and then it swings forward and throws those spores out into the air. Very cool. So in the seedless plants, spores are the way that we get our offspring out into the world, which is okay. We're going to see a new innovation here in the seed plants. I suppose before we get there, we need to talk about some of the internal structure. All right, so we were just talking about sporangia, places where spores are made. In the female, let me back up and tell you, I don't have a slide for this. In the males, the spore is retained. So that pollen grain is the gametophyte. We know the gametophyte comes from spores. The pollen grain develops inside the male individual. So the spores never leave the male individual, nor the female. The spore stays put. It's not dispersed at all. What gets dispersed is the gametophyte individual, which is crazy. Okay. In the female, we've got a little bit more complexity. The female sporangium, the place where the spore is surrounded by some layers of tissue. On the left, this is a sporangium in a gymnosperm pine tree. This is the sporangium in an angiosperm on the right. Okay, so here is, think of this for now as the spore. It's actually the cell that would divide to give us 4 spores. One of them goes away. We just are left with one viable spore. So for now, in our introduction, think of this as the spore. So we've got one spore inside this sporangium. It's surrounded by tissue, and it never leaves. It never leaves. The surrounding layers of tissue around the sporangium are called integuments. Okay, so the spore is retained inside the parent sporophyte. The parent sporophyte makes the spore and then holds on to it. The spore, like it's always done, germinates into a gametophyte. In our other plants, we've seen this gametophyte as a free living individual out in the world. Now that is not the case. The gametophyte stays inside the parent sporophyte. And one great benefit of this is that it allows for fertilization to happen inside the individual, so inside controlled conditions rather than sperm just swimming out in the world. So this allows for internal fertilization. We've got our female sporangium, layers of tissue around the outside called integuments. We'll look more closely at this next week, but this is your introduction. Oh, I should tell you that this new structure has a name. It's called the ovule. The ovule is a sporangium with integuments surrounding it. And we have heterosporia again, two kinds of spores. So this is really the megasporangium with the megaspore surrounded by integuments. And all of that together is called the ovule. All right. They're seed plants, so we know that they have seeds. What is a seed? We've talked about this already. The downside of being an embryophyte, that the sporophytes have to live in the same places as the gametophyte. The upside, of course, is that the sporophyte gets this initial boost from the gametophyte, which is already established. Seed plants overcome this limitation, this downside of embryo retention, by packaging the embryo with resources in this seed. So we've made this sporophyte gametophyte connection mobile, portable. Okay, so now we can, instead of throwing our spores out into the world, we can actually throw our baby sporophytes out into the world, our diploid organisms out into the world. So here's the seed. We've talked about the parts of the seed before, but that was outside of this context of the life cycle. So let's come back around to the seed. Three major parts. On the outside, we have a protective coating. We call it the seed coat. This comes from those integument layers around the megasporangium. We've got this new tissue. You've heard this word before, endosperm. This endosperm tissue is produced by or derived from the female gametophyte. So we talked about the endosperm providing nutrition. Now you know where that tissue comes from. It comes from the gametophyte. And then the third really important thing in the seed is the embryo, the new plant. So we still have this connection, this embryophyte connection. The embryo being nourished by the endosperm. So it's not the whole living photosynthesizing gametophyte individual, but it's still resources from that gametophyte individual. And this is all surrounded in the seed coat, so we can throw it out into the world. It can just be dispersed. So the embryo, the new plant, is free to move into a different location. It'll still have some resources, and it'll have this protection of the seed coat. I've already kind of said these, but here they are in words. Benefits of the seed. We've got our embryo with its food supply surrounded by a protective coating. This allows for dispersal. Embryos can stay dormant for a long time, and the seed coat is protective. We can also enlist the help of animals to disperse the seeds. So now we are here at this end of our continuum. We started with a free-living gametophyte and a totally dependent sporophyte, and now we're on this other side with a free-living sporophyte and a totally dependent gametophyte. Our seedless vascular plants were this intermediate place with a free-living sporophyte and a free-living gametophyte. Initial dependence of the sporophyte, but that's overcome shortly after the sporophyte gets its own roots.